Hello everyone, and welcome to Group 10's presentation for molecular machines. Today we will be discussing the protein dynein and two designated articles for support. Before diving into specific mechanisms and studies, it's helpful to have a general overview over what dynein is. Dynein is a multi-subunit motor protein found in close proximity with microtubules and acts as an essential factor to promote forceful movement of a cell and movement within a cell. More specifically, Dynein utilizes ATP hydrolysis to invoke a power stroke conformation that physically moves itself, and with it, cell cargo and retrograde transportation. This is in contrast to the opposing motor protein kinesin, which is generally involved in anterograde transportation. Cell motility, vesicle transportation, and mitosis are just a few examples of essential cell functions that rely on the presence of dynein. While this protein was hypothesized to have some ATPase function, the mechanism of the power stroke was unclear. This presentation will dive into the research methods and its results used to derive a better understanding of the power stroke mechanism of dynein.
The protein dynein was first discovered in 1965 as having adenosine triphosphatase activity. It was isolated from a genus of cilia, Tetrahymena pyriformis. After employing density gradient fractionation, a separation technique that separates based on density through centrifugation, two protein subunits were discovered, 14S and 30S. Enzymatic techniques revealed that 14S and 30S were related as monomer and polymer, respectively. Electron micrographs revealed that the outer fibers of the cilium consist of the 30S subunit, which itself is a linear polymer of globular 14S subunits. This was supported by evidence suggesting the 14S and 30S subunits had similar enzymatic properties in both sedimented at 10S. The globular units can be seen in Figure D, and how they make up the structure of 30S can be seen in Figure C. Finally, 14S and 30S both presented high specificity for the binding of ATP. The electron microscopy techniques are discussed in depth in the following slide.
Dynein C is a purified isoform of Chlamydomonas reharsi flagella, and its specimens were applied to carbon grids treated with ultraviolet light, rinsed with MMED buffer, and stained with 1% uranyl acetate. The structure of Dynein C was found through transmission electron microscopy, also known as TEM, along with single particle analysis. In transmission electron microscopy, the electron source emits a beam of electrons, and the condenser lens focuses the electrons in a fine beam by controlling the energy intensity. The beam passes through the specimen, which must be very thin in order for electrons to get transmitted. The objective lens focuses the transmitted electrons to produce an intermediate image, while the projector lens magnifies the image. The fluorescent screen at the bottom of the microscope converts the electrical signal to a visual signal to help us visualize the TEM image and diffraction patterns. Then, single particle analysis is used as a processing technique to analyze multiple TEM images to create an image that has a more accurate and easily interpretable feature. This figure is an actual image of Dynein C by transmission electron microscopy and single particle analysis. Dynein consists of four major parts: the head, stem, stalk, and linker. We will talk about the linker in the next few slides. Every dynein is a complex that contains between 1 and 3 heavy chains and has a molecular mass which is greater than 500,000 daltons. A heavy chain folds to form the globular head, which contains the arrangement of domains. The first domain, which is AAA1, that is closest to the end terminus, hydrolyzes ATP that causes a conformational change and leads to the power stroke. The stalk was proposed to be an antiparallel coiled coil that binds to the microtubules. The stem binds to the intermediate and light chains that attach cargo to dynein. These structures are essential in the function of dynein and its transportation.
The pre-power stroke of dynein, also known as the ADP vanadate conformation, as shown in the highlighted figure to the right, is the structure dynein has prior to movement. Vanadate is used to lock the protein into a stable pre-power stroke conformation. It is absent in the post-power stroke conformation, and the transition from ADP vanadate to APO state is analogous to product release and power stroke. It has a roughly circular head, and imaging reveals that the stalk and stem are both flexible. Flexibility of the stalk produces a continuous range of motion, but in the pre-power stroke conformation, the stalk is consistently curved as shown in the lower image. The post-power stroke will be discussed further in the next slide.
Post-power stroke conformation is represented by the nucleotide-free or APO version of Dynein, highlighted in this middle image shown here. Notably, the dominating structural difference between these conformations is that the APO molecule is more compact, and there's a stiffening of the stalk compared to the gradual curve as seen in the ADP vanadate conformation. And overall, we can see the head and stem are significantly closer in proximity. This shift in the stalk and stem positions is shown in the merged image on the right. While the proximity of the stem and stalk increases, there is no observed change in the actual length of these respective domains. Therefore, the origin of movement must lie between these two structures. Shifting our focus now to the globular head domain, which is shown below in these lower images, imaging reveals the APO molecule has a more segmented head along with a central stain deposit, which is labeled here. I'll break down this portion in the next slide. To reveal the origins of this central channel, investigators isolated rare, long-stemmed Dynein molecules, which are distinguished with a near 90-degree angle, which is shown in this bottom left image. These unique molecules reveal the stem's true connection point to the head, which is known as the linker, which is noted with an L. The release of ATP separates AAA domains 1 and 2, as shown in this image on the right, and this is a conformational change and reveals this inner channel seen in the staining deposit. So together, these findings promote the linker domain as an integral part of the Dynein power stroke.
Electron microscopy determined that both conformations of Dynein have the same general structure. However, the post-power stroke conformation is more compact. The power stroke of Dynein results in firstly the loss of products, then the stiffening of the stalk, and finally tension in the stalk that is used to move a load. Stiffening in the stalk can be seen through the decrease in average angle and length between stalk and stem. Figure G shows that the angle decreases from 160 to 136 degrees, while figure H shows that the length decreases from 45.3 to 40.4 nanometers. Stiffening of the stalk had an additional experimental result in exposing the base of the stalk, confirming to researchers the stalk was composed of two alpha helices that formed an anti-parallel coiled coil, which both have ATP-sensitive microtubule binding domains at the tip.
Analysis of the reviewed articles has led to the main conclusion that the power stroke of Dynein is unique, although uncertain. However, the second article does propose an intricate mechanism based on the changes in conformations as seen by the single particle analysis study. Initially, Dynein is attached statically to the microtubule until the binding of ATP to the AAA1 subunit promotes release of Dynein from the microtubule. During this release, you can see that the linker and head are allowed to freely rotate and extend to a position for reattachment further down the microtubule than its initial position. The hydrolysis of ATP, which most likely occurs in the AAA1 subunit, induces binding of Dynein back onto the microtubule and results in a tight binding between the microtubule and the binding domain of the stalk. This tight binding is found to promote a conformational change and rotates the AAA1 through 4 head subunits, and as mentioned before, once ADP and phosphate are released, they are now considered to be in the APO state. Thus, the Dynein protein is contracted. Specifically, the AAA1 subunit and linker are found to be coupled and promote a stiffness. And as these two become more rigid, the Dynein protein is slowly contracted or pulled across the microtubule. It's estimated that with no cargo, this power stroke is able to translate movement by roughly 15 nanometers. And while this article does propose a unique power stroke mechanism of Dynein, the concept of a power stroke is not uncommon and can be found in many other popular mechanisms, such as the popular actin-myosin power stroke.
Image classification via TEM and single particle analysis reveals major structural changes in Dynein C and provides the groundwork for the proposed mechanism of Dynein's power stroke. Comparison of the ADP vanadate and APO molecules reveal the flexibility of stem and stalk, and rare Dynein molecules reveal a linker domain connecting the stem to the head. Post-power stroke molecules are more compact, which is predicted to originate from a change in linker orientation and the coiled coil stiffen. Together, these structural changes create the foundation or mechanism of the Dynein power stroke. While these rare long-stem Dynein molecules were crucial in the discovery of the linker domain, their physiological significance remains unclear, and elucidating this may reveal more accuracy in Dynein's mechanism of force. Finally, two-way communication of the microtubule binding domain down to the stem is known, but the mechanism of this communication isn't clear. Thank you for watching, and please stay tuned for the Q&A.